Hello students. So let us discuss today about the lungs, the structure briefly and the working of the lungs with respect to human beings. So we will draw a diagram. So this is the voice box or the larynx, and this is the trachea, and the trachea in the chest cavity will divide into two bronchi. And this will be divided into, there will be two lungs. These bronchi actually go inside and they divide into first primary and then secondary and tertiary bronchi. And this part over here is the diaphragm. So this is the left lung. We need to show that the trachea has got cartilages, so here we are showing that the first part is the larynx, but before that is a pharynx, and of course the nose. So the first part will be the nose, then the pharynx, then the larynx through which the air comes inside. Then there is a trachea. The trachea has got many C-shaped cartilages. That means the cartilage is incomplete and it shows movement all around, and therefore it is placed in such a way that the trachea gets support. So even if we are compressed, our chest cavity is compressed, still the trachea don't collapse. It's very important that it doesn't collapse. The trachea will then divide into bronchi. So there are two bronchi, left and the right bronchi. Then these bronchi will divide into will keep dividing inside the lungs as primary, secondary and tertiary bronchi, and then these will divide into bronchioles, and ultimately as terminal bronchioles. In all except the terminal bronchioles is presence of cilia internally. Also that they keep producing a lot of mucus. Also that they keep producing mucus, and therefore in the entire respiratory tract, whatever particles we may have taken, pollutants we may have taken, so all the particulate matter including pollutants, they are all expelled outside, expelled out by cough. So this is the natural reaction of the body to throw out whatever the particles have entered inside, and there is presence of cilia in the trachea, in the bronchi, and in all the bronchi excepting the terminal bronchioles. Excepting the terminal bronchioles, cilia and mucus is produced everywhere in large amount. Cilia is produced, and the cilia help to drive out particulate matter along with the mucus which is generated. Then we have the two lungs. So you can see over here in this position is that there is a notch over here which is on the left lung, notch in left lung to accommodate heart, and the lungs are covered by pleura. So let us show this. So everywhere over here the lungs are covered by pleura. This left lung will be divided into two lobes, and this right lung will be approximately divided into three lobes. So these are lobes. Two pleural membranes. There are two pleural membranes, and there is a fluid inside them. Which allows easy breathing. So one of the pleural membranes is attached to the chest cavity, other is closely attached to the lungs, and therefore in between the fluid acts like a buffer, and it allows easy breathing to take place. Infection of the pleura leads to pleuritis, and the left lung is divided into two lobes. The right lung is divided into three lobes, and below the lungs is the diaphragm. Now let us talk about ventilation. Ventilation means movement of air over the lungs. movement of air to over the lungs. In this case, the lungs. If it was gills, then if the animal is in water, when water moves over the gills, that's called as ventilation. So naturally over here there are two steps. One will be called as inspiration and the other is called as expiration. During inspiration, chest cavity expands, and the chest cavity expands because of the ribs. The ribs move outward and downward. Therefore, this creates a negative pressure inside. This creates a negative pressure inside, and this negative pressure therefore draws atmospheric air inside. So I'll repeat this. The inspiration, this is because the chest cavity expands. The chest cavity expands because the ribs move out and down. Also the diaphragm on the dome shaped structure that moves down. At that time the stomach moves a little outside, therefore the diaphragm moves down. Due to this there is negative pressure created and the atmospheric air moves in, atmospheric air moves inside. So this is like a suction pump. If you recollect, in frog it was a pressure pump. This is like a suction pump. This inspiration is because of the diaphragm and the ribs, the rib cage. So all that is allowing this inspiration to take place. And the reverse will take place in expiration. So during expiration, the ribs will move inwards and upward, and the diaphragm will once again get back to its normal position, and therefore the air inside the lungs moves out. Now, although these words are not mentioned here, so the expiration is release of from lungs to outside, release of spent air. Now the air that we breathe in and out during a regular breathing, normal breathing, this is called as tidal volume. And sometimes you can breathe, take a very deep breath. So that is called as maximum inspiration or maximum inspiratory volume. So tidal volume is about 0.5 liters. Maximum inspiratory volume will be much more, say around 2 liters. At the same time, apart from the tidal volume, there will be a maximum expiratory volume, which will be about 1.5 liters. So this total is called as vital capacity. So this you can see is about 4 liters. But even if somebody takes a full deep breath right from beginning to the end, still there will be some air which is always going to remain in the spaces of the bronchi or in the bronchioles actually, which are not expelled outside. So this accounts for residual volume and dead space. This is about 1.5 liters. So that will be the entire or the total capacity of the lungs. But just these are important to know, especially what is the meaning of tidal volume and what is the meaning of vital capacity of lungs. Then let us come to the next part is, we said that the structure, what is there inside the lungs. Inside the lungs, the terminal bronchiole, it will end up into tiny sacs which we call as alveoli. So how did the terminal bronchiole come by? See, the bronchus divides. There are 2 bronchi. Each bronchus will divide, primary, secondary, tertiary, like a tree, like the trunk of a tree. It will divide, and then these tertiary bronchi will again divide into finer tubules. These are called as bronchioles, and ultimately divides into a terminal bronchiole. So in this terminal bronchiole, very tiny one, then it will end up into a bunch of grape-like structures. These are called as the air spaces, and each alveole is very thin. It's a very thin space, but it will be spherical space. And this alveoli, they are surrounded by blood vessels. In this case, it will be by a network of capillaries. So it is this alveoli. If you recollect, in the introductory topic to lungs, we said that whenever there is exchange of air, that respiratory surface must be thin, it must be moist, it must be vascular, and it must be naked. And here the alveoli are performing all the functions because it is thin, it is highly vascular, it is naked, and of course it's allowing for exchange of gases at this point. So this we can draw over here. There will be one layer of cells surrounding the alveoli. Let's draw a bigger diagram. So if this is the alveoli, so there are cells over here. These are epithelial cells. These are epithelial. This is the air space inside. This is layer of cells. And among the important cells is that there are cells of alveoli secrete surfactants. What is a surfactant? Surfactant is something like surf or soap. Surf or a soap. And this surf or soap helps to reduce surface tension, and it allows the lungs to breathe. It allows the lungs to breathe. That means we are inspiring air inside and we are expiring air outside. During this process, the lungs must be highly elastic. So the elasticity of the lungs is very crucial. So it must allow the lungs to become big, and it must allow the lungs to collapse and become small. This elasticity is because of surfactants. I just want to mention that during this period of the SARS-CoV-2 infection or the COVID-19 infection, one of the things that happens, or one of the symptoms that takes place, is that the virus is able to destroy production of the cells that produce surfactants, and that's why it becomes so difficult for the person to expire. During COVID-19, that the person is unable to throw out or push out the air which is already there in the lungs, and this therefore does not allow the next batch of air to come inside. So that is because of the cells of the alveoli. So coming back to this is one spherical ball-like structure of the alveoli. There are many which have been shown over here. All these are the alveoli, and this is the terminal bronchiole. So we will label this the terminal bronchiole, and these are the bunch of alveoli. And each alveolus will be spherical like this with an air space inside, and they are richly supplied here by blood vessels. I am just showing this like a circle. So these are all the blood vessels. So this is not just one blood vessel, but capillaries are there, completely capillarized. So between the blood and the air, therefore between blood and air, only two membranes, one of the alveolus and one of the capillary, and because of this, exchange of gases becomes very easy. Remember that in the lungs, the pulmonary artery takes to the heart deoxygenated blood, and the pulmonary vein deals with oxygenated blood. That means the artery comes in with, so actually the artery comes into the lungs with deoxygenated blood, and the pulmonary vein takes to the heart oxygenated blood. Hemoglobin is the respiratory pigment which is very, very important or it works as oxygen carrier. So there is a molecule of heme plus 4 globin chains. Globin is the protein and heme is the porphyrin molecule. So heme plus 4 globin chains. Each molecule of Hb can carry 4 oxygens, but generally they are saturated with saturated with 3 oxygens, because they have to distribute oxygen to all the hemoglobin molecules, and therefore they get saturated with 3 of them. And it is because of this hemoglobin that we are able to supply oxygen to all the different parts of the body from the lungs. So from the lungs, this hemoglobin is there, which is very important molecule. It has a crucial role as a respiratory pigment, as a carrier for oxygen. It also, of course, carries carbon dioxide and hydrogen as HHB and HbCO2. So Hb combines with oxygen to form HbO2. Hb can also, in the reverse condition, can combine with carbon dioxide to form HbCO2. And Hb also combines with hydrogen to form HHB during inside, so that a buffer is formed to remove the acidity. This is called as hydroxyhemoglobin, this is called as carboxyhemoglobin, and this is called as oxyhemoglobin. So all these molecules are formed in a reversible manner, because hemoglobin can not only accept oxygen easily, but it can also easily dissociate from oxygen. So it easily dissociates. When we say easily dissociates, it means that in the place in the body where there is less oxygen, it will easily dissociate to release oxygen. So this is how exchange of gases will take place, and so this is coming back here. This is the alveolus. From the alveolus, from inside the alveolus, O2 goes into the lungs. The reason is simply diffusion, because there is more oxygen concentration in the air over here, less in the lungs, so O2 diffuses in, whereas CO2 from inside simply diffuses into this alveolar space. Now let us deal with transport of CO2. We just dealt with transport of oxygen some time back here, transport of oxygen. This is transport of CO2. See, CO2 plus H2O, these are metabolites in tissue. So lot of CO2 is produced in the tissue because of metabolic reaction. So this combination of CO2 and H2O does not take place much in the tissue. So what happens is that they have to enter into the red blood cell. So this is RBC. So CO2 and H2O, they enter inside. And once they enter inside, they form something called as H2CO3. So H2CO3 is carbonic acid. And this carbonic acid now dissociates very quickly inside the RBC. I am referring to inside the RBC as H plus and HCO3 minus. Now this H plus combines with HB to form HHB. So it is getting buffered. But the HCO3 moves outside, negative ion. And once one negative ion moves out, there is a negative ion that moves in, Cl minus. This is called as chloride shift. And this HCO3 will now combine with, say, potassium or sodium and will form either, say, for example, NaHCO3 as salt of sodium or KHCO3, and goes up to the lungs and again reverts back to this and will go out as CO2. This is required to buffer the blood. And this phenomenon that I explained to you now is called as chloride shift. So nearly 70% of CO2 is transported in this manner as salts of, say, for example, NaHCO3. This is how it is getting transported. Say about 20% or so, 22% of CO2 is transported as HbCO2, carboxyhemoglobin, and very less as just in plasma as dissolved CO2. So maximum amount of CO2 is by this method, okay, where it is being transported as bicarbonates of sodium and potassium, and some amount of this is transported as HbCO2. So this is how it moves. So we have now considered all the points in this chapter.